for being here still after 2 days. And yeah, I'm Leonard Vennenberg. I'm from the Netherlands, Utrecht, actually representing the mainland of Europe here. So thank you for being so inclusive. So thank you for to invite me. It's not so nice to put me as moderator after a professional moderator. But on the other hand, I hope the news will not run so late here as what we've seen in the previous discussion. And I will not be so cheap to compare you with any person from films or whatever. Actually, Michael Douglas, I was expecting him to compare Matthew Cuman. He's not here, is he? No, I was expecting him to compare Matthew Cuman with Elvis Presley, but he didn't. So, all right, the debate, 25 minutes, starting from now, 25 minutes. We have two, beautiful ladies here. Is it still allowed to say that? I think it is, yeah. Pam, do you want to introduce yourself? Forget about the dame. It's an independent discussion. We're fair to each other. We're not all dames, okay? So just introduce yourself as a person, okay? Oh, wait, you can't hear it. This is terrible. This is not my choice, not Pam's choice. Okay, thank you. That was great.
So I'm Pam Shaw from the University of Sheffield. The dame is the most teasable thing imaginable. I've even had people sing into my voicemail, there ain't nothing like a dame. Anyway, I'm going to be making the case today that before we have cocktails of therapies, we need targeted treatments with some evidence of efficacy.
Okay, the next, I think we're getting music. Yeah, surprise, no music for Angela? Yeah, for Angela? Yeah. Oh.
Clarify, that was you sexy thing.
I believe in miracles. Song of hope.
Okay, thanks.
This is going to be true.
So,
yeah, who are you? And what's your opening statement?
so
You have to sit down, sit down. Yeah.
okay, so I'm Angela Genge. I'm a professor, not a dame, from Montreal at the Neuro in Montreal. I have been compared to Jodie Foster, one up on Jeff. I think she makes more money. I think he spends it on his ex-wives, but I'm just saying. My case is that why wait? This is currently considered a heterogeneous disease. Heterogeneity needs cocktails. Let's not wait for the miracle. Let's get started now.
Okay, great. Yeah. Okay, I think we're almost ready. Pam already wanted to change her slides, but that's not allowed anymore. So we'll start the poll, but we first start with the official debate. Please shake hands, okay? I'm completely independent. I won't vote, but yeah, okay, good. We try to do that afterwards as well. Please sit down. Okay, the voting. Is it clear what they vote for? Yeah. Okay. All right. Targeted. Okay. Oh, wow. Okay, work to do, Pam. Oh, I shouldn't have said that. Okay. Yeah, 39 versus 60, 1% undecided. Okay. Yeah. Oh, no, time is up. Okay, 5 minutes for Pam, 5 minutes for Angela. Yeah, don't worry. I won't time it. And then we'll start. Then they have an opportunity to ask some questions to each other, and then the audience, it's your turn. Okay, please prepare your questions. All right, Pam, 5 minutes starting now.
Very good. So I'm making the case that targeted treatments are the best way forward as we are at the moment. So I'm a barrister, directing you to my point of view that targets are best. My learned colleague on my right here, Professor Genge, is the barista making the case for cocktails. So we all know that ALS MND is a complex process. Even when we know a genetic cause, there are at least 11 or 12 different things that go wrong to contribute to motor neuron injury. The therapies we have at the moment, Riluzole tackles excitotoxicity, Tofersen tackles SOD1 MND, 2 other FDA-approved drugs haven't come through in recent phase 3 trials. But Riluzole has modest effect, not surprisingly, because as far as we know, it's only tackling one of those damaging mechanisms. So the foundations of my barrister case are threefold. Firstly, targeted genetic therapies have generated the most important advances in MND therapeutics to date. This is the array of genetic therapies that are targeted to specific causes of MND. And SOD1 I'll talk about a little further. Fuzz looks very promising. The C9 trials, as Kevin showed us, have unfortunately not, we're back to the drawing board for those. The ataxin genetic therapy for sporadic MND also was terminated. So targeted genetic therapies, what has happened with SOD1 is we've gone right upstream of all those damaging mechanisms to lower the expression of the protein that's causing the disease. And this is a beautiful example of translation from basic science in the laboratories. This is a pair of brothers from the same litter. The mouse running around looking fairly normal has had SOD1 genetic therapy. His brother is the control mice. So showing in cell models, animal models, that the treatment works. You've heard a lot about the Tofersen trials. So beautiful target engagement, lowering of the SOD1 protein in CSF, dramatic lowering of neurofilaments starting at 3 months. But the clinical benefits took longer to show, but they were there by 12 months. So the ALS functional rating scale, respiratory function tests, muscle strength by handheld dynamometry, an excellent slide because that's showing improvement in the group that had been on Tofersen from the beginning. And that's what some of our patients were showing us. So the human impact of this targeted therapy, this is the first patient in the UK that went on to Tofersen in a wheelchair, could just bear weight with 2 crutches at the beginning. When he was in the trial, going up his garden steps, which he hadn't been able to do for 2 years, in the open label extension, walking around his house with no walking aids. So dramatically beneficial. But also, I would argue that a targeted therapy can address multiple disease mechanisms. And I'll show you one example we've been working on from Sheffield. So NRF2 is a transcription factor that controls the antioxidant response, sometimes known as the programmed cell life pathway. And that system is dysregulated in MND, both in cell models, in human tissue, and so on. So that antioxidant defense system is not working properly. And if we could get around that and increase the expression of this protective agent, we would add to the effect of riluzole on excitotoxicity by tackling all these points of damage shown by the green arrows. So we've screened molecules that would upregulate that NRF system. We've taken it over several years through cell models, different mouse models of MND. It's got orphan drug designation by the EMA and FDA. We've manufactured it because it's a drug that hasn't been used in humans before. We've done toxicology testing in mouse and non-human primates. And interestingly, Fight MND helped us to gather all that data. In the 7 minutes that Leonard's allowing me, I can't show you all the details, but it activates not only NRF2, but heat shock factor 1 pathways as well. So, and I won't show you all the data, but some highlights. So in vitro, in human cell models, it has neuroprotective effects. It tackles TDP43 proteinopathy, and it works in both SOD1 and TDP43 mouse models. So these are human astrocyte motor neuron co-cultures. Astrocytes from MND patients are toxic to co-cultured motor neurons, and our best NRF2 activator improves motor neuron survival. It gets rid of the TDP43 proteinopathy, this 35 kilodalton fragment that accumulates as TDP43 proteinopathy is happening. And in animal models, it improves neurophysiological parameters of motor neuron function measured by compound muscle action potential. So my third argument is that before a cocktail of drugs could work, each individual therapeutic agent must be shown to hit the target and to give at least some signal that it's helping. And there's a whole array. We did a review just a year ago of all the small molecule and cell-based therapies that are coming forward into clinical trials, tackling multiple different mechanisms. And so we've got a whole pipeline of targeted treatments that can eventually be put together into a cocktail, but we're not ready for that yet. So are you going to vote for enlightened targets to tackle the motor neuron injury in MND, or cocktails in the dark? I rest my case.
Thank you, Brian. Sit down. Sit down. Yeah, sit down. Angela, your time. Starting now.
No, I have to have my slides first.
Okay, 5 minutes.
And I think that was more than 5 minutes, just saying.
It was
So,
exactly 5 minutes,
an
but you can
amazing-
start now.
and it was
Oh, no,
not,
no, not yet.
so thank you, Pam, for an amazing
Yep, there are your slides.
statement with very clear science behind it, and finishing your talk with the word cocktail. So we're going with cocktails. What will, no, can I use this? What can I use? Oh, there we go. I think we need to really consider what we enjoy in life. Don't we? Don't we all really need to focus on a really nice cocktail? So I actually didn't figure out what you want to drink here in Australia, so I stuck with my Aperol spritz. The joy of having a complete cocktail rather than the sadness of having one after the other after the other. My argument here is we already like cocktails. We've already started treating in a cocktail fashion. In some countries, people are using 3 approved drugs. United States, for instance. In other countries, we're still aching for riluzole, but we've already started down this path for a disease which, if it was simple, it would have been solved in the 40s. We know it's complex. We know we have multiple pathways. Each need to be targeted one at a time. one at a time would take forever. We have to be able to pull them together. So I'd like to share with you an example of where cocktail therapy really made a difference, along with philanthropy, along with money, along with working together. So it's a giveaway the music, because this is Freddie Mercury. At his peak, heading Queen, he could do nothing wrong. He was phenomenal. The fact that everyone here knew the song tells me what a legacy he left. Here's a photo of him just before he died. Freddie Mercury is one of the many tragedies of HIV/AIDS, and I started my career in medicine taking care of these guys, mostly men in the wards I had, although we had a substantial number of Haitians in Montreal, and they just died. And we tried to take care, we added antibiotics, we treated CMV, we treated TB, but it was a fatal disease. And through those late 80s, early 90s, I also worked in a neuro-AIDS clinic, again, because of the type of infections these guys had. And so I was part of that wave that started with AZT and DDI and DDC. These were the kind of drugs we are now working with in ALS. It's a shorter span of time, but it's the combination of your riluzole, your Nuedexta, your Radicava. Effects on survival, slowing of progression, but we don't know when that next one is going to come that blows things out of the water. In HIV, they were using all those drugs, and then the protease inhibitors came along. It wasn't fancy in my mind now. They simply added the protease inhibitors to the AZTs and the DDIs that were doing some benefit, although not a dramatic benefit. And what did they find in 1996? They had a Lazarus effect. Was it the best drug ever? That perfect drug has come since 1996. Over time, they're down to much more sophisticated treatments for HIV/AIDS. And now they have that perfect drug. But to deny people that treatment for 20 years until you get the perfect drug by using your cocktail, you are actually allowing the patients in front of you to benefit right away. Are there other examples? Well, this is the combination that was that first breakthrough combination. And I saw those men. I saw them basically get out of their beds and start to walk and gain back strength and gain back muscle and deal with survival guilt because their best friends had died 6 months before. But we want to have that. We want to set the stage by adding drugs to what's already approved to get to that cocktail. By the way, Jeff, it was TCH346 from Novartis, and it was Zaliprodin from Sanofi. Two great stories on not to combine or how to combine. Another quick example of combination therapy is the traditional and still used treatments in oncology. This is a snapshot of the CHOP regime. Those of us who started treating patients in the 90s probably wrote those scripts as we were interns and residents and et cetera, doing our rotations in oncology. Clearly successful. Clearly now in oncology, there's targeted immunotherapy and gene modifying therapy, but there's several decades between when people survive because of cocktail therapy and now where we're much more directed. So this is an example from a paper. Dame Shaw was, I think, last author on or first author, one of, and it's a cartoon, but it's a critical cartoon. Look how many pathways we've already identified as affected in different patients. Maybe all in the same, maybe at different times of the disease, but different patients, different pathways that we can target. These are the number of therapies that are out there. This is a snapshot of the number of therapies. There's on clinicaltrials.gov, I think somebody quoted 100. And let me tell you, these small biotechs, they don't have the money to go all the way to the end of phase 3 and find a failure. They're looking to know very early on in the process whether we stop and switch to another. But they're doing all of their drug development on the background of already having the equivalent of AZT and DDC. They're doing it on the background of our riluzole, our radicava, our Nudexta, and the other drugs that people are bringing on now. So at NCALS, this is how many posters there were. A whole list just on new drugs in development. And this is the rest of the list. But you don't have to fly all the way to Sweden to see evidence for cocktail therapy. You just had to listen to the talk earlier today. And that talk was beautifully done and demonstrated right in Australia that cocktails based on standard of care or added into standard of care are the way to go with great evidence. He can repeat his talk after we finish just to reinforce how good the work is in Australia. So I believe in the science that has demonstrated that this drug is not just a tuberculosis. It's just not one single cause right now. Maybe we'll find that ultimate cause, but in the next 5, 10, 20 years, patients are getting ALS, MND. We have multiple pathways. We need to look at cocktail therapy. So my final slides are, this is a Canadian, you can tell by the clothes, Olympian from a couple Olympics ago that did win gold. Really strong guy. He could tackle the world by himself. However, here's an 8. They do it faster. They move times are better. And I believe that with the evidence we have to remember a heterogeneous disease, cocktail therapies. Thank you.
Okay, thank you. Well done, both. You already sound a lot more hopeful and positive than this very depressing panel before tea break. So let me ask you, please sit down. Both have to, no, you have to sit here. You have to listen to me, otherwise I kick you out. Pam, so targeted therapy. So why were you so negative in the previous panel discussion about curing the disease? Well, actually, we could treat all the SOD1 positive carriers before they develop the disease. Isn't that curing ALS?
I think, as someone said earlier, I think it was Kevin, tofersen is not without its side effects in 7% of patients. So my feeling is we move step by step. And the next step in SOD1 is identifying the very earliest stages of disease, either by an increase in neurofilaments or somebody developing fasciculations, a little bit of weakness. I think we're not ready to treat healthy, asymptomatic people, and we don't know when their disease is going to start. So
yeah, but I think the question from this news moderator guy was, do you think we will develop a cure within the next, what was it, 20 or 100 years? I mean, that we can do it in the next 5 years.
So I believe we'll make step-by-step progress in slowing disease progression.
All right,
I don't think we should be talking about cures.
Thank
But
you.
may I argue something to my
Yeah, I'll ask
colleague?
one question to Angela because I want to be fair. Angela, so why have we not followed the AIDS and the oncology and actually tuberculosis path? I mean, we're still doing this mono drug trials, but also the preclinical research is all, one drug that only tests one drug at a time. Why don't we look more at synergistic effect?
So we are, for 2 reasons. One is regulatory. Regulatory are forcing us into this path of monotherapy. It's not monotherapy, it's add-on therapy. So we're already doing cocktail in regions that have approved drugs, but regulatory are forcing us down this path. Now, there's a whole movement out there to look, to develop through AI and other, mainly AI innovation, digital twins and avatars, so that patients never have to go on a placebo to generate the data that the regulators want. But essentially.
The second one is? You had two reasons. I want to speed up a little bit because
What do you want? What was it?
you had two. One was the regulators, two.
And two is the community. The
Yeah,
community sets a very high bar for response to therapy. In the HIV/AIDS era and in the oncology field, they don't set the same bar. And in some ways, setting a lower bar has enabled them to move faster. Even if you look at MS, the first drug, that was a very low bar. The beta interferons, we didn't ask for
okay, time is up,
revolution.
time is up. Very nice, well done. So the community is also the people here, is it? Yeah, say yes.
Not
It's maybe
everybody
not
here.
popular, but
Some of the
Yeah.
people here.
some people here.
So I
Not
would
you, but some
argue-
people.
I would,
All
ar-
right, Pam, you can ask a question now to Angela,
so
and then Angela can ask you a question as well.
So I would argue, Angela, that those effective cocktails for HIV, tuberculosis, certain types of cancer have been based on targeted drugs that had some effect on the disease and then put them together and they have a more major effect. But it wasn't just a cocktail appearing out of the blue. It was based on putting together drugs that hit the target, had some effect on disease. And in HIV, you can measure the viral load and, white blood cell subsets. In oncology, you can obviously measure tumor size and so on. So it was targeted therapies that had some effect, different mechanisms of action, and then put them together and you get a stronger effect. So
Do I
your
get to respond?
cocktails were
Yeah, is that
based
still the question?
on
Yeah.
good targets.
Yeah.
So my response to that is, if you actually look at the effects of these drugs independently, they were modest. They are what we're finding in our phase 2 trials. And so they were prepared to take the risk of combining drugs that had modest effects. We're already doing that, guys. We're already doing cocktail therapy because of the ethics boards and for good reason. But none of those drugs independently had a huge effect. They all had the modest effects that we're looking for in phase 2 and went ahead and did that bold study.
All right. All right, thanks. And so now Angela,
Have a question.
no, Angela has a question.
Oh, I
A
have
question
one.
for Pam, yeah.
So,
Okay.
yeah, I'm just
Fire
getting
away.
mixed up here. Okay,
Yeah.
so my question is really a more like a patient advocacy question, actually. If you wait until we get that one targeted drug, which we don't know when it's coming, and I was involved in the SOD1, the tofersen development. Like, I knew about it before we started the phase 1. I started screening patients in 2014, all comers, for their genetic mutations in the clinic, because I knew it was coming. And it took until, when was the first approval for one drug?
September. Oh, for
No, the
tofersen,
first approval for tofersen was in
Oh.
the
April.
US,
April
and-
2020.
23. And the drug, so that's 13 years.
You have to be more to the point to your questions.
Okay.
Otherwise,
Well, no, so
what's the question to Pam?
why wait so long?
Yeah.
If we have drugs
Because-
that have a modest effect, why
But-
wait so long?
But Angela, where are the components of your cocktail? Where are the drugs with modest effect, apart from riluzole, possibly edaravone? Where is what are the ingredients of your cocktail?
Well, that's a
Because
great question.
we know that phase 2 trials may look hopeful, but then they don't deliver in a larger scale pivotal trial.
Do I get to-
80%, Jeff said.
So,
and
yeah,
failed
and I
that
think I'm
stage.
gonna,
So where are the ingredients of your cocktail that you want to give patients?
I think Jeff alluded to it. I blame it all on Jeff, you can tell. It's in academia. There are people running trials, investigator-initiated trials, small trials, because that's what they can afford, in academic centers around the world, and are showing positive results. I think we just had one from that veterinary drug.
12 patients.
That's right, but the response was there, and that drug actually moved neurofilament.
Okay.
So,
Yeah. no,
so
yeah.
there, I'm
in
cut off.
three-
Okay,
In 3 patients.
I came close to finishing the
Yeah.
sentence.
All right. Thanks. Thanks so much. Questions from the audience? Yeah, go ahead. Yeah, Lucy. Who's running with the microphone? Am I? No, I hope not. Yeah, go ahead. Go ahead. Go ahead.
I'll talk loud.
Yeah.
So essentially we should say add-ons is different because we've all agreed and Pam has agreed things are going to be added on. And in the meantime, Pam is developing the single target. So if it's a cocktail, you've got to bring the cocktail together with all the ingredients. So you throw in 3 drugs, whether they're academic or not, and you give that, and yes, it actually has some effect. How do you ever improve that effect? Because you don't know which one of those 3 of the cocktail actually worked. And so you're left always with a very modest effect, whereas you could actually then take the one that really worked and modify it to make it, to drive it to the right place in a better way. So you just cannot build cocktails, whether academic or industry, without testing each one of them and then enhancing them.
So I guess that's to you, Angela.
Yeah, so, but, no. No.
She works for Novartis. huh?
I would counter that even, and I would look again back, first example is HIV. They were all modest effects. It was the protease inhibitors alone also had a modest effect. It was when the 3 were combined that they had the effect. The argument really is we have some evidence in phase 2, 1B or 2, biomarker only, because we've all said you need a clinical response to get a drug approved. We get 2 or 3 molecules all showing that in phase 2. Do we really need to run each one individually as a targeted therapy all the way through a very expensive pivotal, or can we just combine the 3 and move the pivotal based on the different target engagement markers, the different pathways they're affecting?
The viable load is a good marker, and maybe then you can, that's it, but it's not, we don't, neurofilament is not that yet.
But there are many, many other biomarkers in development that are, many companies now are matching their drug to a target engagement biomarker. Depends on the drug. Depends on the program.
Yeah. Yeah, Michael Douglas.
So you make it, you make a convincing,
Absolutely love it,
Yeah.
Jeff.
you make a convincing argument. My dad was a good actor too. You make a convincing,
To who are you?
wait a minute, you make a convincing argument.
Angela or to Pam?
No, sorry, this is Angela, that the combination in HIV with a small effect, and you're relating that to in unbelievably tiny ALS trials. I want to go back to real facts. When those drugs were studied in HIV in those small and what you call modest effects, how big were those trials? Were they 12 patients or were they 200 patients?
It was at the moment, at that time, they were huge compared to.
So
what
that's an important point. You're blending things. And when we see trials, and we see them all the time, a company says, look, I did 12 patients and sorry to use 12, but we've seen lots of them. And I think this is positive. 12, we know ALS, it's incredibly variable. And if that was small trial was 200 and it was a modest effect, now we would combine it. But
Yeah.
don't do that mixture. That's just bad science.
Okay.
Yeah.
Any, oh, yeah, you have to answer. Yeah, sorry.
Do I have to answer?
No, you don't have to.
Okay,
Yeah.
but I think we're making,
Can, if-
so
if you
the
want.
argument for cocktails, other than cocktails are fun, is, and I'd love to hit the magic bullet every time with all of our trials. But the argument is, do we really need that long-term 500 patient trial in each of the three drugs that we see some target engagement or some exceptional response in phase 2? Do we need to wait through all of those trials to get to the cocktail therapy?
We
And
actually
I would
have,
argue there's a path.
we actually have Julian Gold here, the AIDS expert. Yeah. You want to say anything about, you want to help Angela a little bit or?
He wants an open label extension for his trial. That's what he wants.
Yeah. Is she okay with the combination treatment, how it started with HIV?
I think that in HIV, to be fair, the problem with using one drug is that HIV developed resistance to that drug reasonably quickly.
All right.
And
Yeah.
then by having the combination, you got around the development of resistance. So the principle of combination was very important. But I think in ALS, it may be that the pathology is actually a bit different. But I do agree that having a combination from the beginning is very important. In HIV, the problem was that the drug companies didn't want to collaborate. They wanted to have the magic drug, they wanted to make all the money themselves, and it was only the academics and the clinicians, and particularly the AIDS activists, that forced the companies to collaborate. And I think that's going to be one of the issues in ALS as well, actually getting these companies to work together.
Yeah, that's actually a
Okay.
good point.
It
Okay,
is a really
yeah,
good point.
thanks.
Are we voting now? Or you
No,
want to-
Is there one question for, we have to,
I had a question.
is there one question for Pam?
No,
No?
My
No, there's no
question's
question for
clear.
Pam? Okay, we're going to vote again.
Well, I want to point out that Australia does combination, as you saw. I'll bring back my slide.
Yeah, okay. Okay, now you can
Yeah.
say something too if you want. A final statement. We have to finish up, but I don't want to rush you. But I think we're going to
I
be kicked
think-
out.
I think now that well, let's see the vote first.
No. Well, there's nothing after the vote.
I think now that we've rehearsed the debate arguments, we're not that far apart. I think combination therapies or a single therapy that tackles multiple mechanisms are the way forward, but we've got to be careful about the ingredients of cocktails, and they have to have good scientific evidence behind them before.
Okay,
we
stop talking. You're
treat
already voted.
patients.
Yep.
Yeah.
Oh, you gained some. Votes. All right, good. So there are no winners in ALS yet. So please, it's a tie. It's tied. Well done. Okay, thanks. Come on. Yeah, they're still friends. So thank you for the questions. I just want to say, as a community, we shouldn't give up hope to find a cure for ALS. So, I mean, I did not agree with the previous session. The hope is the most important part we all have for our patients. So keep on working for a cure for ALS. Okay, thank you.